All right. Hello, welcome back. This is going to be the first post-lab video I'm making. I'll try and go over what we went over in lab, just as a little recap of everything that we've gone over. A few things I want to say before I dive into the recap. Great job, everybody, for wearing your masks. I think everybody was wearing masks, so I didn't have to tell anybody to put a mask on. So that was a good job on y'all's part. Second thing, make sure you check on TopNet, see which section you're in. There were a few people who showed up to the wrong section. Make sure you're checking TopNet and go to the right section and go to the right time frame too. So make sure you're showing up at the time specified by the first letter of your last name. So that way we were able to socially distance properly. What we went over in lab this week was basically an introduction to zoology and a little bit of a review from past classes. So what is zoology? Zoology is just the scientific study of animals, and this ranges from animal psychology or their behavior to the form and function of organisms, what makes that animal move, what allows it to circulate blood through its body. And so structure and physiology is covered in the dissections of this lab. I mentioned this earlier during lab, but I try and fit as many function facts as I can, so that way these dissections become more appealing to the individuals who want to go into the medical field. So a large part of biology is taxonomy, or just classifying organisms. And so that's what the next slide shows. It shows the breakdown of that hierarchical network of organisms and how we describe them. So I gave the slide of the dog-maned leaf beetle, which is native to Kentucky. I mentioned that everything in this course will be in one of two things. It'll be in the domain Eukarya, or it will be in the kingdom Animalia. So obviously, since zoology is the scientific study of animals, everything from here on out will be in the kingdom Animalia, or animals. But kind of lesser known is that everything here will be in the domain Eukarya. So there will be this nuclear envelope that surrounds the nucleus. We go through lots of phyla, which is plural for phylum. In fact, we start our next phyla next week, and you all should be watching the video on the phylum Porifera that I'll post this weekend. We go through a few classes, such as Insecta, the insects. We go through a lot later on in the semester when we start talking about vertebrates or arthropods. We don't really go into orders, families, genera, or species, just because it takes too much time to go into that level of detail. So even though there are a lot of really neat organisms, we just can't fit it all into one course. So, the next thing I talked about was using and making dichotomous keys. And so dichotomous key is a tool that we use to identify something that's unknown. You can use dichotomous keys to identify what's wrong with your car if you break down. They actually make those sorts of keys. But the keys that we'll focus on in this class are biological in nature. And so we're always trying to identify an unknown organism. You have to account for the geographic location of the organism. So if you're identifying or you're trying to identify a specimen that you collected in the Barren River, you can't use keys that are specific to Europe or Africa or Asia. You have to use keys that are specific to that geographic location. You also have to account for the sex of the organism. If you remember the dichotomous key that was in your lab manual, there was the mallard and the gadwall. So when you use the key to key out both birds we had in class, you had the mallard and then a female mallard. So even though you used the key correctly, you got the incorrect answer when using the female mallard. I did mention, or I should have mentioned, that males and females look nothing alike in most cases. Males usually be much more bright because they compete for females, whereas females can be kind of drab and boring because they don't have to compete for mates. The other thing you have to account for when using a dichotomous key is the life stage of the organism. If you're using a key for, I mentioned dragonflies, if you're using a key for dragonflies, the larvae look nothing alike like the adults. So you have to use a life stage specific key, meaning the distinguishing characteristics are not found in larvae, they're only found in adults. So if you use a key for the adults, you won't get very far. When you're making a dichotomous key, you always start out with something broad, some sort of broad statement, and then you work your way to more specific details, so that way you can start narrowing down which species you could be referring to. You also order the specimens based on, you order the, don't know what I'm doing. You order the statements in each couplet based on the level of importance. So I don't know if you can see my mouse or not, but this first statement right here, the fully extruded Ediagus with single elongate anterior dorsal finger-like membranous tube and lateral view with distinct posterior ventral lobe and paired anterior dorsal lobes. That's the most important statement in that couplet part. Right behind it, it states distribution larger streams of the interior plains of the Midwestern U.S. That's not as important as the anatomical statement is. So when you do this, the reader will be going through this key, and they'll see this first. They'll check to make sure it matches the organism, and then they'll read the second part. And if that matches, they might have that organism. They can read through the second part of that key or of that couplet. So they go through 17 that might match. They go through 17 prime. If that doesn't match, then they know they have 17. That's isoprola bilineata. If it doesn't match either of the statements, if it has a fully extruded Ediagus without a single elongate finger-like membranous tube and Ediagus with lobes otherwise, you move on through the couplet and through the key to another until you find another species. So that's how dichotomous keys work. You guys might be using those next week. We'll see on that one. The next thing I covered in this lab was, as it says on the screen, a really brief review of Biology 122, 123, where I discussed phylogenetic trees. And so phylogenetic trees are really just working hypotheses. They change all the time based on the data that we have available. And so what they show are the evolutionary relationships between groups of organisms or organisms. So you can have these really specific ones, such as the one to the right, the one from 2016, or you can have these really broad ones that only cover phyla, such as this one here. When making your phylogenetic tree, you always start with an outgroup. This is how you root your phylogenetic tree. It is a group that is a common ancestor to all the groups past it, but it doesn't include most of the common characteristics that other groups have. So I gave the example of the Cranoflagellates. In my next video, I'll talk about the Cranoflagellate Theory, but basically, in a nutshell, Cranoflagellates are single-celled organisms. They're colonial, so they can work together. And it's hypothesized that those that did work together became more successful and continued to work together, become more successful until they became an actual single unit, single organism. And so that's when they became multicellular, and that's when they started forming these complex structures that we consider sea sponges. So everything from periphery on, is what we consider the phylogenetic tree, while the Cranoflagellates are the outgroup of that tree. The next thing I went over was a polytomy, or an unresolved node. And this is when there's some level of uncertainty between groups where we don't truly know what the common ancestor was. And so in order to display that we don't know this and that there's some level of uncertainty, there will be two branches stemming from one node. The next one that I went over was a homology. And so homology is when there's a trait shared within different groups of organisms due to having a common ancestor with that trait. So there's the example of Echinodermata and chordates. They both have deuterostome development at some point in their lives. Another example is multicellularity. So everything in this phylogenetic tree, except for the Cranoflagellates, are multicellular. So everything here is a homology, besides the Cranoflagellates. I love to give the example of flight being an example of convergent evolution. So flights evolved several times in the course of evolutionary history. Flights evolved with sauropsids, or the dinosaurs, with the pterodactyl, and other flying dinosaurs. It's evolved with birds, so really modern day dinosaurs. It's evolved with insects. Most insects have evolved some form of flight, but very basic insects did not have that capability. And then flights evolved in vertebrates, or not vertebrates, in mammals with bats. So really primitive mammals can't fly, but bats can. I also gave the example of an eye. Eyes have evolved 3 times that we know of over the course of evolutionary history. One was in mollusks, so octopi and snails. They all have an eye, which is similar to what we have, but unique in its own regards. Vertebrates, so we have an eye, rabbits, other mammals, reptiles, they all have eyes. And then the third time the eye has evolved is in arthropods. So crustaceans and insects have their own version of an eye. Basically, any trait that is that works very well will be selected for over time, even in groups that don't have those capabilities yet. And so this trait becomes so prevalent that it occurs many times over history. So history has a tendency to repeat itself, and that's especially true with evolutionary history. The next thing we went over was assessing relatedness in phylogenetic trees. And so we talked about the difference in relation between Coelomorpha and Rotifera, and the Coelomorpha and Mollusca. And so when you're trying to see how related two groups are, you count the number of common ancestors or nodes that they share between them. So if you look at Mollusca, that has 6 common ancestors between Mollusca and Coelomorpha. If you look at Rotifera, Rotifera has only 4 common ancestors between Rotifera and Coelomorpha. So Rotifera is more closely related to Coelomorpha than Mollusca is. You could do the same thing with chordates. If you count the differences between chordata and nematoda, or let's do something different. Between chordata and Annelida, we have 1, 2, 3, 4, 5, 6 differences. If you want to know the difference between Porifera and Annelida, you have 1, 2, 3, 4, 5, 6, 7, 8 differences. So technically, by count of common ancestors, the chordates are more closely related to Annelids than Porifera is. I mentioned this a lot, but phylogenetic trees are working hypotheses. They change all the time based on the data that we have. So genetic data is very cheap, relatively speaking. It's quick. You can have organisms, you can have their DNA read in labs. You get the data back very quickly. So this causes trees to change a lot. there's the example in this slide of arthropods and anilids, so segmented worms and insects and crustaceans being grouped together based solely on segmentation. And so this is being grouped solely on morphological data, and then genetic data is what separates them out. And so arthropods are actually more closely related to nematodes, and then anilids stay where they are. So yeah, that is a very quick summary of what we covered in the lab this week. Be on the lookout for the next lecture video that I'll post. It'll be sometime this weekend. Make sure you watch it. It'll be much more in depth than this one has been. So next week, be sure to wear your mask. Always have it with you, and wear closed-toed shoes. Remember to take the lab quiz. So it'll be posted Sunday at 11:59, and then it'll run until the hour before your before labs that day. So 12:40 on 12:40 on Wednesday and 12:40 on Thursday. So, alrighty.